So again, thank you for all being here. I forgot to introduce my co-chair before we started the plenary session. So David, my apologies. But David Balshaw is here from the NIHS, who will be co-chairing the whole day with me with this symposium where we really want to address basically how we come from the concept that Chris beautifully introduced to basically how do we bring that in practice and where are we. And so we're very happy with having a large line of speakers today. As Chris basically already indicated, if we look at the chronic diseases, then we can basically, based on migration studies, based on twin registries, basically discern that a large part of the disease burdens that we see have some kind of environmental component. And this is from a paper from Walter Willett in 2002 in Science, basically making the argument that that could be somewhere between 60 to 80% where the environment in conjunction with genes actually play a large part in the disease burden. If we then go a little bit further, we basically can ask a question, what do we know of these new and environmental risks? And this is basically borrowed from the Global Burden of Disease project. This is the paper from Francevar in 2015. And what you can see in this picture is basically the total burden of disease, basically by these two concentric pies, where if we look at the colored pies, basically we can see that here about 1.3% of the total burden can be assigned to environmental and metabolic factors. This is basically 12% to environmental factors and so on. So maybe from this modifiable part of the disease burden that we have based on the GBD, we can see that we probably at this moment can attribute probably about 40 to 41% to known risk factors, of which the environment plays a large part in itself. However, if we look at this gray circle around it, is basically the 60% of the total proportion that we have no idea what the environmental drivers are. And I guess this is basically the challenge that we face today, is basically how do we actually identify those unknown factors and better quantify the disease risks that we have. You could come up with several reasons why we don't know what we don't know. And one is that basically the current risks that we currently have, we basically underestimate because we actually don't have the right exposure proxies and therefore basically have attenuated risk estimates and therefore under ascertain basically the burden of disease. It might be that we actually are using inappropriate risk models where we often basically look at adult life, cumulative exposure and relate that to disease, where we actually don't really take into consideration the dynamics of the exposure or the time windows of critical exposure, as Chris already pointed out, being that somewhere in early life or during early adulthood, where the exposures actually can have differential effects than they have later in life. Of course, it's also true that we probably are dealing with a lot of unknown risks, basically risks that we haven't identified until now that are out there, which we basically are yet to discover. The third point is that we basically could also argue that some of that may be completely purely stochastic and there may be not a reason to really see what is driving that. So as Chris, and I know that Chris used another definition which was not his own definition, but I wanted to give credits to Chris by putting Chris' definition up here, which Chris pointed out in 2005, basically realizing this difference between the investments in genomics versus in exposure science and trying to get actually the environmental component measured accurately by saying that exposome is a science of impact of the environment broadly defined on health and disease and refers to the totality of exposures from conception to death. It's based on technical developments and external measurements, such as sensors, which we term basically the external exposome, which you can divide again into the specific or the general external exposome, and internal omics measurements or the internal exposome. And so this is basically the onset of this concept of the exposome. As Chris pointed out, and this is also for junior researchers, not to be despaired if your paper doesn't do anything after you publish it. This is basically the number of papers that actually came out after 2005 on the exposome concept. And basically, till 2008, there was the only paper that was there was the first paper by Chris, and then 2009, there was another paper picking up the term. And it really changed indeed when Steve Rappaport, Martin Smith, and others in the US basically starting to pick up on this concept, bringing out the fact that indeed, if from a public health standpoint, if the environment is that important, why are we not making this investment and actually characterizing that environment much more accurately? A National Academy of Science meeting was conveyed, and after that there has been a exponential growth on papers on the exposome. And I stopped counting in 2014, where we're up to about 100 papers that really was describing how the exposome potentially could be used. And I say the word potentially here deliberate because what we really are at the time now, and I think Chris was completely right by saying now we are in the teenage years, where we have to make the switch from the concept of what we could do to really see how can we actually implement that in real life studies, and what do we learn when we try to do that. After basically the definition of what the exposome was, there has been a lot of discussion about what it exactly is in the literature. I did it on Scrabble. If you do it on Scrabble, the exposome gives you 20 points if you're interested. You can go back tonight. You can actually get 66 points with the word exposome if you're lucky on the Scrabble board. But basically, after basically the definition that Chris gave, there was a lot of discussion about, well, if you say the totality of exposure, and Chris pointed this out already, that it's really impractical to be able to measure the totality of exposure from birth to death. And so we have to make choices. We can actually measure parts of it. Other arguments that were made by Steve Rappaport is basically, well, we have done basically a lot of research where we had candidates, and we really have to move to agnostic approaches, and how do we do that? But by saying that, by really measuring basically internal markers of exposure or reflections in the biological system that may relate to exposure, other researchers basically made the remark, well, that is of interest, but if you're not able to really bridge that and make the link to external exposures, you won't be able to intervene, and you won't be able to actually devise successful public health interventions. So basically we need all those elements to be there. This session and this symposium that we have here today is basically not to debate about what is really the definition of the exposome. Both the chairs will not allow any discussions about that definition. I'm just pointing this out that, you know, you can approach this problem that we are facing really to identify that environmental component of disease risk from different angles, and different aspects are going to be important in identifying those and relating those to successful public health interventions. That's what we really want to address today. Basically, how do we go from these concepts, from these ideas that we have, to really see how we can use that in practice. As I said, there are very strong opinions, and I just put this email out that I received in 2012 when I was giving a keynote on the exposome, basically saying that I was a bad boy by actually addressing the exposome, and that it was really not what we should do, and it was in conflict with everything that we had learned in exposure science. I think the times have this changed in the sense that we now realize that the exposome definition is not just basically a holy grail, or this is the only way to get to a solution. It actually is putting, again, environment and environmental exposure assessment and how do we actually find these things at the forefront of the work that we do. And I think that's what we have to take away from this. And now we actually have to change and see how we actually move forward by implementing these concepts. If you look at the definition that Chris gave in 2005, I think you can pick out three elements. One is basically, when we start to implement it, it's basically we have to think about study design. If we really want to do life course, it means that we actually have to do life course, and we have to be able to actually address the fact that exposure is dynamic, and that it actually changes over life. And for that, you may need repeated samples or biomarkers that reflect past exposure, or external models that actually are dynamic that can address that. So that we actually can look at what are the exposures during critical exposure windows, during basically conception to death. The other element that is in that definition, or the several definitions that have been given, is basically how do we get to improved exposure assessment, either from the external side or the internal side. And there's a whole array of different techniques that one could use. You could think indeed about biomarkers or omics markers, which Chris already gave some nice examples from. but there's also sensors and wearable technologies that can be used in this framework as well. GIS information, which is getting more and more available at a much finer spatial scale every single time when a new satellite goes into space. We now all carry mobile phones or portable computerized devices, so to speak, which allows us another way of collecting data, which we can explore in this framework as well. And of course, there's the improvement of just conventional measurements by questionnaires and databases that we actually can use. It's not that new technologies, and Chris was completely right by saying that this field should not be driven by the technologies, it should be driven by the questions that we have and use the best tool that we have to address that question. Of course, when we actually move forward, we actually have to develop new statistical methods and bioinformatical tools to actually make sense from the large amount of data that we actually collect and the dynamics of the exposure distributions that we actually collect. What we will have to deal with are basically large numbers of environmental exposures that we have, which are correlated and protracting. We have to deal with the fact that many of those, because we don't have strong priors, have basically weak associations and low priors. So we are running the risk that we're actually generating quite a significant amount of false positives, and we have to carefully think about how do we move forward when we start implementing these techniques. So I just borrowed this slide from Chris from his keynote, which he shared with me, so I was able to put this up very quickly now. I think when we go through the day, indeed, these are the questions that we want to address, basically. We have to remain focused on the question and on the task that we have put ourselves to, is basically identify that environmental component of disease. How do we do that? What are the tools that we can use? And what are then basically the lessons that we learn? It's indeed a phase of taste and see. Today we'll be hearing presentations of the large projects that have started in Japan, the US, and Europe that actually started to implement at least parts of this theory into practice. And what do we actually see? We are in the very early phases, so this is a time that we are going to make mistakes, and this is a time that we are going to learn. And today is basically to actually show those and discuss with you all here about what are the ways to go forward, what are the things that we may have to change when we actually move forward. So I won't go through the whole program, but just to basically give you an idea what the program of the whole day is. Basically, in the morning, we really want to focus on how to build actually studies that use this exposome context. And we will go through all the large efforts that are going on across the world to basically see how people have taken up this concept and now trying to implement that into active research. In the afternoon, we basically want to show some proof of principle studies, just to show that there are some lights here in some of this vast amount of space that we haven't explored yet, where there are some bright stars where we can actually learn something about how this may actually work and where we can see how this can be used towards the future. The last part of today, we really want to focus on this problem that we are going to face, basically, when we generate that amount of data. How do we actually come to valid statistical inference? How do we come to causal inference? How do we integrate bioinformatics? How do we integrate prior information? How do we integrate basically evidence from animal experiments and from in vivo testing into basically the data that we're actually generating within these large prospective studies now? So that's the program, and I was inspired by Chris about quoting Winston Churchill, and Winston Churchill had another saying, and he said basically, this isn't the end, this is even the beginning of the end, however this is the end of the beginning. And I think that's where we are. We're actually now at the end of the beginning, where we have the first basically proofs of principle, the first efforts that take forward, and this is really just the early stages where we actually have to move forward and to be brave and actually move this field forward. So with that, I just wanted to introduce the first speaker of today, Martine Vrijheid, who's working at ISES Global, which used to be CREAL, which is a name that is probably more familiar with a lot of people. Martine is leading the very large HELIX European project, which is trying to basically measure the early life exposome. Martine has had a very successful and rich career, being trained here in the Netherlands, then moving to the London School, and then moving to the agency, and then moved to CREAL and has been really instrumental in setting up large birth cohorts and looking at early life exposures, and now actually moving that forward into an exposome paradigm. So with that, I would like to invite Martine to come up. Thank you very much.
I'm not sure how to start the presentation.
It will come up.
It will come up. Okay.
To start.
So let me start by thanking the organizers of this symposium for putting this together. I think it will be a really exciting day. And thank you, Roel, for the excellent introduction. It makes my talk a lot easier. So I was asked to give some earlier observations of HELIX. For those of you who don't know, HELIX is an FP7 project. And we started in 2013 and we finish next year already. So HELIX has as main focus the early parts of life, critical periods. In early life, we focus on pregnancy and childhood. So we have some repeated measurements, but all in this very early time frame. And our main aim is to try and identify the environmental components here in early life and how they affect child health and development. A second very important focus of HELIX is on multiple exposures. So we try and integrate exposures from each of these different exposome domains. Obviously there's overlaps between these domains, but we've really tried to put a sort of more holistic, more global exposure view together by focusing on the three areas of the exposome, the specific external environment, as we call it, the general external environment, and then trying to integrate that with the internal molecular environment. In terms of multiple exposures in the general external environment, now HELIX is a project on environmental pollutants and environmental contaminants and some other environmental factors. We cannot possibly look at everything in terms of environment, but we have a list of what we call outdoor exposures. We call them outdoor exposures mainly because they're determined by where people live. They're measured through mainly GIS methods, so this is geographical modeling. So we have measures on air pollution, noise, traffic load and density, the built environment, green spaces, and some on UV radiation and meteorological variables. And then, in the specific what we call more the individual exposures, meaning in the exposures that are measured more on an individual level, we have a list of pollutants mainly, measured through fairly conventional methods, biomarkers mainly. So we measure persistent organic pollutants in blood, pesticide phthalates and phenols in the urine, metals again in blood, cotinine in urine, and then we have some measurements in indoor air. We model disinfection byproducts, and we have long questionnaires on diet, physical activity, and other factors. And this is done both in the mothers during the pregnancy and in the children when they're a little bit older, at the age of 6 to 9 years. The third part of HELIX is to integrate the internal environment. So this little part of the picture. And here the focus, well, the aim is to identify biomarkers of these multiple external exposures and to hope to try and identify some common functional pathways through these omics methods. So we have a whole list of omics methods. We have metabolomics in urine and blood. Proteomics is a very targeted assay, the multiplex. We do transcriptomics and microRNAs and the DNA methylation. HELIX is based in existing cohorts, so we thought it made much more sense for an exposome project to build on all the data that's already being collected in existing cohort studies, because it's very hard to start an exposome project from scratch. So we identified 6 cohorts across Europe, where the mothers were enrolled at, well, similar time periods. So the children were at similar ages when the project started. And the parts of the cohorts that we use total about 32,000 subjects. Now, from these 32,000 mother-child pairs, we have a whole lot of existing data. All the previous follow-up data, health endpoints, some GIS modeling, they have stored samples. And because we know where the mothers lived and the children at each time point, we're able to model the outdoor exposomes, all the outdoor exposures, in these entire cohorts. Now, it's not possible to do biomarker work on 32,000 mothers and children, within this project anyway. So we selected from those cohorts a sub-cohort of 1,200 mother-child pairs when the children were between 6 and 9 years old. And in that subset, we did all the work in a very standardized way with common protocols and common questionnaires, and collecting new biosamples in these children. We measured all the new exposure biomarkers in these children in the same way, in the same lab. We did the whole range of omics platforms that I showed you in these samples. And we collected very standardized data on health endpoints, including neurodevelopment, growth, obesity, and lung function and respiratory health questionnaires. And then we added to that some very intensive panel studies. So these are studies nested within the sub-cohort, at least for the children, where we followed 150 of these children very intensively in 2 weeks of the year. And they gave daily urines, they carried personal monitors, etc. And the idea of that was to look in much more depth at the variability and the uncertainties in exposure assessment, and we used some of this data as validation in the bigger cohort. So in terms of early observations, I'm going to just focus on a few fairly descriptive parts of the project. So all our work on exposures and health associations, I will not present today. It's not ready yet to be presented. But we have some early observations on the descriptive work in terms of the exposures. So I'm going to talk about variability in the exposure biomarkers and a little bit in variability in the outdoor exposures and in the metabolomics measurements. And then we're going to look a little bit at our work on exposure correlations and patterns. And the reason for that is to do this type of exposome work, we do need to understand how our exposures correlate and how to use them in our future exposome analyses. So the first point is variability in exposure biomarkers. What we know is that some exposure measures are highly variable. I mean, this is nothing new, obviously. But when you do an exposome study, this becomes more of a problem. I don't know if it's a problem, but it's something that we really need to look at very carefully, because if you have all these different exposures with different scenarios, and this is just a picture of how this could look. For example, in red here, you might envisage the persistent pollutants that go up during the pregnancy and then stay fairly, and during breastfeeding and then stay fairly stable. You might have a heavy metal exposure during the pregnancy and then again fairly stable. And then you have exposures that might vary a lot seasonally, and you might have exposures, and here we mainly think about non-persistent pollutants that are just variable all the way along at very low doses. So if you try and measure an exposome at any one point, you may or you may not get a good estimate of any one of these exposures, and you don't know. And if you put that all together in an analysis later on, that may give you problems in interpretation if you do not take account of the different uncertainties in these exposures. So one part of our work is to try and really characterize the variability, especially in the more variable exposures. So here we focus on the phenols, including BPA, phthalates, and organophosphate pesticides. And we do that using the data that we collected in the panel studies, using the urines of the children, of the pregnant women, where we have daily urines in the evening, in the morning and in the evening. So we can compare concentrations across 2 different weeks and across days. This is data from the 45 pregnant women where we collected 2 urines per day for 7 days, and this is the type of patterns you see. So from that type of data, we can calculate interclass correlations. For this particular phthalate, this is MEHP. The interclass correlation is 0.42. 5 minutes. And in that way, we can calculate how many samples we would need to analyze to get a reasonably good estimate. And we can do that for many of our pollutants, in the children, in the mothers. But particularly, we see a lot of variability here in the pesticides. Now, what we will do with this type of information, first of all, in this way, we can determine how well what we do in our larger subcohort predicts the exposure in the week before. So it gives us some idea of how good our estimates are in the subcohort. And we can include these interclass correlation coefficients later on in the exposure response models using regression calibration type approaches. So that gives us the opportunity to actually adjust for the differences in uncertainties between exposures. And I think that's important to later on help the interpretation of the exposome models. In a similar way, we look at variability in the outdoor exposures using the personal measurements that were similarly done in the panel studies for many of the air pollution exposures, contact with natural spaces, noise, travel duration, physical activity, measured through these personal monitors and smartphones. So again, we will get from this interclass correlation coefficients, and we will get an idea of how we can attribute the variance in some of these exposures. I don't have time to go into the detail now, but we can see that there's a lot of differences between exposures. Some of them are mainly determined by in which city people live, and others are more determined by the time point of the year. And similarly, we're very interested in seeing how the NMR metabolites vary across the week. So again, using the panel study data, we can look at how the levels of different metabolites vary between time points of the day, between children, and between different time periods. Here we see that there is a large range of, a large difference between metabolites. Some of them are fairly stable, and others vary a lot within the child. So that's the work we're doing on variability. Similarly, I think it's very important to describe correlations and patterns in the exposure. Roel just mentioned this. Correlation structures are acutely important in exposome studies. We've learned from some early simulation work that, for example, the IRAS methods, even with an FDR correction, they don't perform very well when you look at the false discovery rate. And this is mainly due to the correlations within the data sets. So it is really very important to describe the correlations before you actually go on and throw all these variables in an analysis. Also, we know that the patterns of exposure, of multiple exposures, are not very well described, but there's actually very little literature on how people are exposed to multiple pollutants. And we want to know how this varies by country, by region, by social class, by diet, etc. So this is our preliminary work using the IMMA cohort. This is a correlation circus plot indicating the correlations. The colors are the families of exposures. So here we see that most of the correlations are within families of exposures. There's not many lines going across. This is in a way good because it means that when you measure, for example, phthalates, that analysis is not confounded by what happens with air pollutants, for example. And this is very, very basic and useful information. But this could be very cohort specific or very much specific to the population that you're studying. So we're doing similar work now in HELIX to try and disentangle a little bit these patterns of exposures. Here we see many more lines going across. And we need to go much deeper into this data. I think it's because here we're mixing data from 6 different regions, 6 different countries. So it could well be that these correlations are due to the locations of the subjects. So in terms of early observations, these are the messages. It's very important to characterize variability and uncertainty in exposure assessments in studies of multiple exposures. Describing the exposome correlations and patterns is important for the interpretation of exposure effects in light of their correlations to other exposures and to understand the predictors of these high exposure profiles. I think this is essential. And further, that the exposome approach with many exposures together will also, it allows for a much more systematic evaluation of the exposure health associations. If we think what's been done before is in many of these cohort studies, many exposures have been analyzed in separate papers. So one paper on air pollution, one on mercury, one on phthalates, one on BPA, in separate papers. So we're actually, what we're doing already is a type of EWAS analysis, but without any correction for multiple testing, without any type of systematic look. So any type of exposome approach, I think, brings at least an advantage in terms of reducing the false discoveries and the selective reporting compared to actually publishing one single paper per exposure. So I think we're making already a step forward here. At least everything in this way, everything is being published. And next, I would just say watch this space, because we're doing a lot of work trying to look at the effects of the multiple exposures, so the exposome on the different health outcomes, focusing on our 3 main areas of child health. And then we're looking at how these multiple exposures impact, what their molecular imprints are using the different omics platforms. So the questions we want to answer there is, do the exposures act through common molecular signatures, or are they actually exposure-specific omics biomarkers or pathways that we can identify in these, using these omics methods? And then as a last step, obviously, we would like to see if the molecular changes explain some of these associations between exposure and health. And there we get to the meet-in-the-middle approaches. So yes, I hope to be able to tell you more about these analyses next year. And I want to acknowledge all the people who work on this project, because there's many in different places. And they've done an enormous amount of work to get this far. Thank you very much.
Thank you very much. Thank you very much, Martine. We have time for one question, I think. Please state your name and affiliation. There's a microphone just behind you, Jacob.
My name is Jacob van Klaveren. I work at the National Institute for Public Health, and I'm quite active in international advices on regulation. How do you regulate the chemicals? You gave a wonderful overview of what you have measured in humans and what you are potentially measuring in humans, the potential of new technology. Do you also know where the exposure comes from the diet or from the environment? For example, yesterday we had a beautiful presentation from Mark Kennedy in the Euromix project, and he put together all the chemicals and he looked what this exposure is coming from. Do you have any idea with your data?
We have questionnaire data. So what we're going to do is look at how these exposures cluster together, and then also at the predictors of some of these exposure patterns. So, the different dietary factors that might underlie the multiple exposure patterns. These are questionnaires, so this is what the subjects have reported to us, but we have questions on many factors. Yeah. Diet, smoking, how they use their outdoor environment, physical activity, you name it. We have a very long questionnaire, and that's the type of data we will use to then try and make sense of the exposure patterns. I don't know if that's going to match with
Yeah,
what
yeah,
you're
well,
doing in
the-
Euromix,
yeah.
but we could...
Yeah, we can discuss
could
it
think
later,
about
but
that,
yeah, we
yeah.
can think about it
Yeah.
because there will be a lot of windows of exposure, time frames, noise,
Right.
but there's a lot of research
So we will
needed.
have the biomarker data and the questionnaire data to
Yes,
look
yes,
at.
yes, exactly.
This is...
Yeah, good.
We have time for one more question, certainly, so go ahead.
I was wondering whether there were any socioeconomic, psychosocial factors that were considered in the number of cohorts that you were...
Yeah, they are. I
Yeah.
mean, yeah, again, they come from questionnaires. And one of the ideas is to look at the socioeconomic patterns that underlie some of the exposure patterns. Well, this is work that's ongoing. So, yeah.
Okay, again, I would like to thank Martine for a wonderful presentation and then to move on to the next speaker. Thank you very much. Our next speaker is Shoji Nakayama. Shoji works at the National Institute for Environmental Studies in Japan. And Shoji has taken on the Herculean task, really, to start a very large cohort in Japan called JAX. And although his presentation is, we do as much exposome as we can, I think he's one of the really leaders in this field and actually has done much more than anybody else in this field. Shoji spent some time at the EPA and then went back to Japan, and I'm very glad, Shoji, that you're here. So go ahead.
I really thank Roel and David to have this kind of session and invite me to be here. So here we go. I hate the guy who brought this paradigm shift into the epidemiology field, and then also the guy who designed our study to go for 100,000. This is a nightmare, To do the exposome for 100,000 is kind of, yeah. Anyway, our study, Japan Environment and Children Study, JAX, is a government-led longitudinal birth cohort study involving 100,000 mother-child pairs. We didn't expect to get this much, but somehow we did. So we had to go with the 100,000 kids. So we have main study and sub-cohort study. Main study involves all the participants. We mainly so we collected biological samples during pregnancy and also at birth from kids and mothers. And then the father's samples are just by chance. And then we administer questionnaires, and then we're going to follow all the kids until they reach 13 years of age. But this is no way just to end at that 13 years old. But this is just the period that we got secure the funding. And then for the sub-cohort study, we selected 5,000 subjects randomly from the main study, and then we do more extensive exposure assessment and as well as the health outcome assessment. And we have another series of studies, like adjunct studies that are conducted by the participating institutes with the extramural funding. So study areas spread across Japan from north all the way down to Okinawa. The actual study place is here, but including some area that was affected by the 2011, earthquake, and then following, nuclear power plant accident. So here's the Fukushima, and as Miyagi, heavily affected by the tsunami. And recently we had the earthquake in Kumamoto area, and one of our station got, the broken ceiling, and then the next morning we came in, all the computers are, wet, Then so we had to replace everything. But anyway, so we're now using about a little less than 60 million US dollars a year. Which sounds a huge budget, but if you divide it that by 100,000, which is about 500 euros or $560 per participant, which is not the big, the money. So we recruited the participants for 3 years, starting from January 2011 to March 2014. And then we started the biological sample analysis in 2014, and then we continued that. So that cost 50 million euros. It sounds big, but if you compare this is from Leo Tresander's paper website, I would say, and we're having a huge amount of economic loss just due to due to this endocrine disrupting chemicals, according to his estimate. If this is just off by 100%, it's still a big number. So our study budget is, so we have to justify that, the use of the 50 million euros a year, and then this is the good example. But anyway, the JAC's key protocols involves main study involving all the 100,000 participants, and subcohort study involving randomly selected 5,000. So some of our Exposome, trial is going into this subcohort study with 5,000, and then some of them are going into this main study. And for the more extensive study, we can only do 5,000, like a home visit, personal monitoring, biospecimen collection, and face-to-face exam, like available sensors. So, if you're gonna want to use $200 like Fitbit, and if you deliver that to 100,000, it's going to be a huge amount of money. So we can do only for probably 5,000. So those are the priority outcomes, all the way from pregnancy reproduction outcomes down to metabolic disorders, including neurodevelopmental disorders, and exposures of interest are not only in the chemicals, but also physical environment, lifestyle, and socioeconomic status. So here's the current status. We recruited 103,103 mothers, which is coincident. So we had about 80% consent rate. That means 80% of the women we approached that area gave us the consent to participate. And then we only got the half of the fathers. That means only half of the fathers come to the hospitals when their partner get pregnant. So we just only get this. And then we had about 100,000 births, live births, let's say. Then we were following them with the questionnaires twice a year, and then we have collected lots of biological samples. And then we started this subcohort study in late 2014, and then we have done a home visit at 1.5 years old, analyzing VOCs, measuring VOCs and aldehyde, PM, and then we collected house dust for the further chemical analysis. And then we have almost done the 2 years old checkups, and then we're preparing for 4 years old examination. So we take, well, at least I take, Chris's original proposal as a JAC's Exposome, which is a wide idea. And then, so this is the definition we think the JACs should follow there. So life course environmental exposures, including all things from the prenatal period onwards. So this definition is very simple but very hard, to implement. So this is I took this picture from Chris's publication. So sure enough that if you have the only one time a huge exposure during in the utero life is different from the intermittent, their exposures. I did the experiment by myself. I just drank a huge lot of in ethanol at one time, which is clearly different, gave us clearly different, effect than just taking a shot of that every day. But anyway, so we need to measure all the exposures, during pregnancy and childhood. And so we have to use all kinds of measures to achieve that. So using the GPS, GIS, this part we're engaged in some modelers to figure out what was the atmospheric exposures, and then I'm now exploring some capability of wearable sensors. And then we're developing personal sampling devices for example, the VOCs and other gaseous compounds. And then we do have to rely on questionnaire and also computer-assisted testing methods, and then as well as the models. And then if we can rely 100% on chemistry, that would be. I'm really happy, for the exposome. If we go into our body, biological samples and then analyze them to figure out exposomes and then early effect biomarkers, and then also combining that with the omics, well, that's just fantastic. But the technology isn't that far yet. So we have to use other measures like wearable sensors and personal sampling devices. So we're now working with the Japanese, the giant, to develop this kind of wearable sensors, and then I'm asking them to add the UV sensors so that we can tell that the kids are on a car or in the car or in indoor or something like that. So, and then. We're going to use the silicon wristband. We're going to have an entire session probably, I think, that the later this week. And then we're gonna ask the kids to wear this and then to measure how much the chemicals they're using are exposing to. And also we're developing passive samplers that can be attached to the school search so that they can bring that to the school and then bring back and then ask them to wear that for a week or two so that we can figure out the time-weighted of average exposure to the volatile compounds. And then we also do care about the other socioeconomic and also psychiatric environment as exposure to, because we know that parents' IQ effect has the effect on the children's IQ. So why I put the caretakers is just we can't now say that mothers or fathers because some of them are not really fathers, and we're collecting the data about the psychiatric status of the parents, caretakers, and there are also child caretaker bonding. That could affect children's neurological development. And then caretakers' IDs and IQs and the community support as well. And then we're going to probably use NIH Toolbox as a computer-assisted testing method. So here's the example. We can use iPad, so we can bring it into the field, and then we can ask the kids to play on this. So. The exposome is the total environment measures. And until we can rely 100% on chemistry, we have to use other measures, sensors and other things to calculate all the environmental exposures. So JAX is looking for any technologies that come up and then we can use. And then I feel this kind of collaboration and consortium of the entire world, research community is very, very important, because Jax is now 5 years old, which is still a younger stage, but Jax is too big to shift very quickly. So we have to be prepared. You know, we have to take a new technology in and test it and validate it in a few years, and then we can implement that in the field. So I thank this kind of opportunity to have the information exchange between a very diverse range of the research community. And here's the challenges. So whether we should go non-target, what's the quality of the non-target analysis? What's the reproducibility? What's the repeatability of the non-target analysis? And then also, so the quality control of all the external measurements, are using the sensors. What's the variability of the sensor technique? What's the intra-individual, evaluation versus inter-individual evaluations? And also, how are we going to do this international harmonization? We use this, they use that. What's the data compatibility? And then, we're collecting council data, but we're not expecting to see so many councils that that is sufficient for us to analyze the environmental causes to the Children's Council. So we have to work with other other cohorts, international partners. So the harmonization of the measurement is very, very important. And the cost, 100,000. So if you just send out the paper or any material of the cost of $2 or so, that costs $200,000, the piece. So the cheap or affordable, I would say, the method is very, very important. So that's why we're gonna have a symposium at the very last day of the conference, calling for the affordable population. The method can be used for the population. So we need affordable chemistry methods as well as affordable external exposure measurement method. So with that said, I'd like to thank all the participating families and regional center staff who are hardworking to moving this study forward, and then also international partners, and then lastly taxpayers to support our study. Thank you very much.
Thank you very much, Shoji, for a wonderful presentation. We have room for several questions, so please come to the microphone. Martine?
Okay, you mentioned international harmonization. Do you have already some idea of which platforms you will use for the biomarker work, and what's already been done and what's already been planned? I'm thinking in terms of, obviously it would be a great idea to later on be able to compare and replicate findings. What is already actually being done or being planned on?
Well, so what we had done was that to develop our QC materials, like for blood and urine and other things. So we have about, for example, 2,000 tubes of pooled QC whole blood samples. And then we work with the German colleagues and French, and then the Chinese colleagues. So we send that out and then ask them to measure, for example, heavy metals we are measuring, like lead and mercury, and then compare the performance of. So they used completely different methods, three or four different methods. But fortunately, for that time, the measurements were very comparable. So we developed the reference materials, and then we're willing to share that with other international communities. And then, so that's one thing. And then also, we think the communication, the talk is also important. What methodology you use, why, and then so because we are young, we can adopt the new methods. But for the existing cohorts, I think the use of those reference materials or pooled samples, QC samples, will be one solution. But we only have the numbers for certified number for metals, but not for the others. So I'd like to call, I'll present at the Uber's colloquium, but I'd like to call for the collaboration to put the verified, the numbers for the other chemicals, so that we can use our pooled samples for it. Thank you.
Yeah, hi, Shoji. Very nice talk, and usually you're so polite, but I said what you really thought this time. I have a question really to you and maybe some other speakers. I mean, one of the fascinating things is these correlations between measurements within this exposome globe that Dr. Patel has been describing, and others. I'm just wondering if across cohorts, I mean, one thing is data harmonization, and are we measuring the same thing using different ways? But would be fascinating also to know how consistent the correlations are between components of this broader set of exposure measures. Do you think there's an opportunity, perhaps amongst the birth cohorts internationally, on at least a subset of exposures to test that hypothesis that there is consistency of these correlations across populations?
That's a very good idea, and then also probably we can look at what's the determinant of there's preserved correlation between the population. If we do that, because we're from Asia, and we may have very different lifestyles than the Western countries. So we may have little different correlation patterns, but if we compare that internationally, probably we'll come up with some determinants of that common exposure, scenarios and stuff. So that's a good idea, and I'm go for it for that.
Just a follow-up, Martine, you basically presented data where you looked at the correlation structure, showing that you had more correlations across families, making the argument that that might be driven basically by differences in populations that you have across Europe, and therefore you get somewhat more stronger correlation between the families. Have you had the opportunity to actually look at stratified by the studies, and do you have a first indication about how consistent that may be? To come back to Chris's question.
No is the answer, because I got that plot yesterday or 2 days ago. But obviously the next step really is to look at this by cohort. We've got 6 cohorts in HELIX. There's the Japanese, there's other studies across the world who might have data that can fit in with this. So I think it's a really important question to look at.
Yeah, I mean, an analogy with genomics, of course, where we have HapMap, it would be good to have something that is called ExpoMap or whatever we call it, to see if there are actually haplotypes of exposure correlations across these different populations. Just time for one short question, Jeff.
Okay, I'll ask my short question then. So it's Jeff Brook from Environment Canada, University of Toronto. I work with the childbirth cohort. Thanks, Shoji, for your talk. So I guess my short question only then is just a cost one. You gave a number of, I think, what was it, $560 per person. What is it if you go down to your sub-cohort of 5,000 mother-child pairs and what you're spending on those? Was that the 560, or that's considerably more, I assume?
Well, that $500 or 500 euros includes all the human resources and everything, and just divided the whole entire cost by the 100,000. But we're spending. How much? I don't. Well, if we take out the human cost, we're spending about, well, not that much, probably for $100 for the testing. But if you take that all the human resources out, but if you add it up, probably 1,000.
But in your more intensive study of your 5,000.
Yeah. Well, for the 5,000, probably if you add up all the human, the field staff costs and everything, probably adds up to 1,000 to 1,200.
Okay, thank you very much, Shoji, for a wonderful presentation. And I would like to invite everybody to take up the challenge that Shoji put out to actually start collaborating and thinking about which affordable technologies actually could be used in studies like JAX. So thank you very much. We would like to move to the next speaker. Unfortunately, Robert Wright, who was going to basically talk about CHEAR, is not here due to family reasons. But luckily, my co-chair, David Balshaw, has been instrumental actually in establishing the CHEAR program and thinking about what could be done within the CHEAR program. And so I'm very happy to introduce David, who works at the NIHS. And I have an old profile, so he was the branch chief of a branch that doesn't exist anymore, but he can tell better than I which branch he's actually leading now. But he has gone from the University of Cincinnati to UNC and is now at NIHS. And I'm very happy to hear about the CHEAR project and what is going on the US side. So thank you very much, David.
Thanks. So yes, I'm the chief of the Exposure Response and Technology branch, which is spun off from the Center for Risk and Integrated Sciences about 3 years ago. So I'm going to actually, because we had a session yesterday focused entirely on CHEAR, I'm going to broaden a little bit to what NIEHS has been doing on the Exposome, and then some thoughts for, to kind of get some ideas going for some discussion later in the day on kind of next steps of where we want to go with the Exposome. So a little bit of historical context. At NIEHS, since I came to the Institute in 2003, we've really been focusing largely on how we change the perspective of exposure from the single analyte, single point in time and space, to really thinking about the reality of how exposures occur as a multi-scale problem, looking at multiple analytes in multiple times, multiple spaces, multiple routes of contact, and kind of blurring the interface. So coming back to Chris's comments in the plenary, where is the border and the interface between exposure and response? How can we use the understanding of biological response to understand the context that the exposure is occurring in, and using the biology to understand the exposure? Ultimately, because we are part of the National Institutes of Health, we do care about the health of the individual, and so we do see the exposome as being a tool. While it is about the exposure fundamentally, it is a tool to understand what are the health impacts of the exposures. So as with everybody else doing exposome work, for about 10 years, we spent the time, we did read Chris's paper in 2005, and we said, yeah, this is brilliant. We really like this idea. It aligns very much with what we had wanted to do. And so we've spent the early years of the exposome, the first decade of the exposome, really focusing on three areas. First is just the conceptualization, wrapping our heads around what is the exposome, what is it about the exposome that makes it different from what we're already trying to accomplish with environmental epidemiology and environmental health science. Building the tools, the technologies, and the capacity in order to be able to do the assessment of the exposome. So we have had the Gene Environment and Health Initiative across NIH, which NIEHS led the exposure biology program, really to understand what is the way we can do really characterization of the external exposome, and think about multiple analyte, multiple time and space resolution of exposure measurements at the individual level. And then starting to think about how we can do some demonstration of the exposome, and I will be perfectly honest in saying that's the last phase, and we're just really now starting to get into this on a large scale. Obviously, as you've already heard from all of the other speakers, this is not easy. This is, there are a number of challenges in implementing the exposome, and it has taken time to get to where we are today. We have to think about, what are the different kinds of challenges and how do these break down? So they're not all technical issues. There are significant sociological challenges associated with doing the exposome, particularly in America, where our research tends to be, frankly, a little bit conservative. And we focus on incremental science and on doing what is doable. There has to be a willingness to do untargeted work. The exposome, in essence, is a hypothesis that there's something that we don't know that is associated with the endpoint that we are interested in. The only way we can see that is to look across everything. And that's anathema. I mean, as scientists, that's not the way we are trained. We are trained to have a specific hypothesis that I know that this is associated with that, and that's what I'm going to study. And so the exposome really asks us to put our training on its ear and think in a very different way. There are the very technological issues of how we do this, both from an analytical chemistry standpoint of how do we make these measurements, how do we take very disparate kinds of data, so if we want to do the true exposome, looking at both external and internal and biological response simultaneously, those are very different kinds of technologies that we need to bring to bear on this, and how we're going to pull all of that data together. The idea of mixtures is a long-standing issue in environmental health. We all know that we're exposed to mixtures, but how do we analyze the mixtures, and how do we make sense of that data? How do we deal with the correlation between? And then the very logistical challenges. So as you heard from Shoji, and from Chris as well, if you want to do a true exposome study, it's going to, from conception to death, is going to take a lifetime. And if you want to have a sufficient power in order to be able to assess any hypotheses, even if you've got 100,000 children that you're recruiting into your study, you're only going to have a handful of incidents of even important high prevalence disorders like autism spectrum disorder. You're not going to have enough to power a study really to look at any individual endpoint. So how do we think about designing a study that will test all of the variability of the exposome, and how are we going to replicate that? Do we do 100,000 cohorts all around the world, and how do we use those to replicate each other? And then we start to think about, what are the different ways we can use the exposome. So the obvious one, the one that we've really focused almost all of the discussion to date, is in epidemiology, and using the exposome as a tool to enhance epidemiology, not replace epidemiology. Environmental epidemiology is not going to go away, but we will generate some new hypotheses that we can bring into, and I think we will empower environmental epidemiology through the exposome. But let's think about some other aspects of the exposome, some different ways we can use this. Can we use it as a tool to enhance mechanistic research and our understanding of the fundamental biology of what's going on underneath the exposure to disease relationship? Can we use the exposome as a tool to enhance risk assessment? Can we revolutionize risk assessment through the exposome? And finally, can we use it as a tool for citizen science? Can we use this to empower communities and individuals to take action to prevent disease and improve the context in which environmental health sciences occurs? So last year, NIEHS got very fortunate that the National Children's Study came to an end, and we were able to redistribute some of the funds from the National Children's Study to establish a new infrastructure, the Children's Health Exposure Analysis Resource, or CHEAR, to really kind of continue that original mandate of understanding how does the environment influence the health and development of children in America. By providing an infrastructure, a monolithic consortium that will really be access to comprehensive analysis of exposures, and we'll get into a little bit of what that will be before. We set up a consortia. We've got 8 grantees. a coordinating center at Westat, a large company in the United States that focuses on coordinating large federal projects. And they are really the public interface. So when you interact with CHEAR, you're going to be interacting primarily initially with the coordinating center, but they're also kind of controlling our internal workflow and processes within the consortia. We've got a network of 6 laboratories that are doing very comprehensive analysis of the exposome. And then we've got a data repository and data analysis center that's creating a number of tools both for the analysis of the exposome, but also for sharing that data and making it available as a public resource that you as an interested investigator will be able to come in and see all of the data that we are generating and use that in your own research as well. So the labs, as I said, we've got 6 labs, oops, go back, around the United States. And we've designed the analysis to really take advantage of the strengths that we already have in traditional biomonitoring. And the same list of things that Martine has said that they're measuring in HELIX, and Shoji said that they're interested in measuring in JEX. We're offering the same kinds of services to the community. Again, this is targeted. This is hypothesis-driven research where you think that PAHs are involved with your endpoint, and so you can come to us and we will provide measurement of those analytes. But at the same time, we're also providing analysis of the exposome, the untargeted analysis, and the unbiased association between exposures and response. And we haven't talked too much about metabolomics thus far, but really it is an integrator. So the way the analysis is done is you're getting a comprehensive analysis side by side of both the exposures and the biological response indicators for the exposure. And then we are providing some targeted analysis of additional measures of biological response so that we can map onto pathways the interface between the targeted and the untargeted analysis. And then we've also designed CHEAR to improve over time. So we've built into all aspects of CHEAR developmental activities that are going to continue to develop and refine and validate methods. As Chris said, this validation of how we do the new measures of exposure really is very critical to building the capabilities in the exposome. So as I said, there's really 3 kinds of analysis that we're doing within CHEAR. And we've designed it so that they feed off of each other. So you have both the targeted and the untargeted analysis, and you can use that to cross-validate. So if you're able to see, you use the hypothesis-driven analysis of specific analytes, and if you see those in the untargeted analysis, it builds the confidence in doing that. And so over time, we hope that we will get at that sociological issue of the willingness to do the untargeted analysis. And you also get additional biological anchoring so that you're able to weight the exposures that you see relative to their impact on the biology and identify those that are most, quote, important of the exposures. So CHEAR is open. We are accepting proposals from the community. I'm not going to go into a whole lot of detail on how that process works. The most important part is to go to our webpage, chearprogram.org, and sign up for an account. There's a complete walkthrough there of how you go through the process of submitting a request for services, undergoing consulting, putting in a full application. If that's approved, then you just ship us samples. It's pretty simple. You ship us samples, we send you data. It should be pretty easy process to get through. So then, let's think a little bit about where we go beyond CHEAR and what's life after CHEAR. So as I said, let's start to think about some different uses of the exposome and how we can build the impact of the exposome on mechanistic understanding. So integration of the exposome as another ohm into multi-omic integration and taking data from the microbiome and understanding the impact of metabolism from the microbiome and the gut flora, the skin flora on metabolism and the response to omics. the idea from Michael Snyder and the group at Stanford on the integrating personal omic profiling and really having a very comprehensive set of analyses that are being done at the individual level to blend the lines between exposure and biological response. Can we use animal models and controlling the exposome of an animal to understand how we can use the biological response to understand the exposure and this idea of biological resilience? So there's a theory, a hypothesis that it's not the actual exposure that is the critical thing. It's the ability to resist the exposure. So biology is actually very robust, and we're not all dying due to all the exposures that we're encountering every day. And so, can you have an assessment based on biological response of the ability to resist the next exposure, which is really the critical determinant of the association to disease. And can we use this to empower the exposome as a tool to understand the associations between exposures and disease? Then risk assessment. We all know that risk is the product of both exposure and hazard. And many of us in the exposure community say, well, don't ignore us when you're doing risk assessment. Both in the United States and in Europe, there is a traditional dominance of hazard and toxicology in risk assessment. And when we do use exposure data in risk assessment, we frequently are doing it inappropriately. We're looking at a single exposure or a single pathway of exposure to look at the risk. We struggle very much to deal with temporality in exposure, and we're using incomplete data to assess the exposures in risk assessment. So can we use the exposome and that comprehensive view of exposure to empower risk assessment? And how can we do that? So I think everybody is probably familiar with the adverse outcome pathway. This has been around for a little while of tracing the individual steps in the pathway of biological response and hazard from the molecular initiating event, the macromolecular interactions of the analyte, all the way down to the organism and the population response. This is a pretty well discussed pathway. And there's been some, just in the last year or so, some discussion around an analogous pathway. We had a workshop primarily led by the US EPA this summer on aggregate exposure pathways. And can we create a parallel to map the cascade from the multiple sources of exposure to multiple analytes all the way down to the target site exposure, and deal with this as a multi-scale phenomena where we can track multiple exposures through multiple sources and multiple pathways to ultimately link up to the molecular initiating event and create a cascade that goes all the way from source to adverse outcome, and a framework that allows us to think about risk very comprehensively and dealing with the complexities of both the exposure and the biological response. So there's again, this is kind of like the exposome was 10 years ago. It's a wild idea, and we need to flesh this out over time. And then finally, the idea of citizen science. So this is my institute director, Linda Birnbaum, really pushes us every day to think about what is the impact of our programs on the individual, on the community, and how can we not only use our science to enhance the understanding of the individual and the community about the value of our research, but can we also use their perspective and their understanding to improve the way we do science? So how do we think about the role of the exposome in this? Many times as we speak with the community and community engaged researchers, many times they're actually more amenable to the idea of the exposome because they know that they are exposed to multiple analytes. They know that that is complex. And they have very significant concerns about diseases that are not explained by our current paradigms. And so they're looking for a different kind of solution. And so as we're thinking about the exposome, I think that we have a challenge as a community. And again, this is not at all fleshed out, but how do we present our information and how do we engage with the community in a discussion about the exposome, the strengths of the exposome, the limitations of the exposome, and move forward with that. And with that, I think I'm probably pretty much done. And just summarizing, challenges kind of going forward, some kind of underarching issues for all of these different applications. How do we make the information available in a format that can be used and understood by the diverse communities that we have to interact with, not only the research community, but also the community. How do we use the exposome as a tool for prevention? It's not just an ivory tower where we're seeking to understand the underlying biology, but we have to put this to use and make something of it. And then finally, as Linda is very fond of saying, and has gotten the NIH director, Francis Collins, to say, we live in a world of gene-environment interaction. That's what causes disease. And you can never change your genes, but you can change your environment. So this really is ultimately, I think, going to revolutionize the way we do prevention. And it's better to not get sick in the first place than it is to try to find a cure for disease. With that, I will finish up. I'll say thank you, and I'm glad to take any questions.
Thank you, David, for laying out the NIHS roadmap for CHEARS and beyond. We have room for questions.
Thank you very much. I'm Jos Bessen from Joint Research Center, European Commission. I think you give us a very nice overview and also some insights in the conflicts that we will probably have in the next few years in the issue of toxicology, animal toxicology versus epidemiology. I think the exposome concept really gives us a nice idea on at least the statement that we should move a little bit away from the animal toxicology. I mean, it's mostly one substance toxicology. As a toxicologist and a risk assessor, I've become aware that exposure is probably much more important than the toxicological hazard. We know that quite well, and we had last week in the Netherlands a really good example on the issue of the use of rubber granules used in artificial soccer fields, where in this broadcasting they invited a toxicology professor. But I thought, what's the news? We know all that the polycyclic aromatic hydrocarbons that leach out of these rubber granules are carcinogenic. Nobody disputes this. But the issue is, what is the exposure? What comes out? So could you give us maybe some insight on how do you think that in the next few years the discussion will move on, where we have, at least in Europe, really tough regulations based on animal toxicology? How could we move forward to, let's say, taking these concepts more into account?
Yeah, so I think we need to be able to convince the community that we can measure the exposures and that doing that adds value. So I think that this is actually a challenge of validation. It's not that we don't have the methods. I mean, the methods for measuring PAH exposures are long-standing and they're well accepted. We just have to convince the risk assessors that doing that really does allow you to say, there is exposure associated with those granules, and that exposure, understanding that exposure helps you place the hazard into a context of what's going on. There certainly are challenges of moving from the animal hazard testing into the extrapolation to humans. Certainly did not talk about it here, but we have a large program at the National Institute of Health, and I know that there are similar programs in Europe on the humanized organotypic cultures, so that you can do some of this in vitro work in a humanized system, so you can get that kind of translation built in, and including the ability to generate those using induced pluripotent cells, so that you can have the genetic diversity built into that as well, and use it. If you couple that with multiple exposures and different kinds of engineered exposure scenarios. So this is what I was alluding to with the animal model, but you could also use it with the organotypic to kind of simulate a complex exposure scenario to look at the interface with risk.
So 10 years from now, we will have a same symposium, let's say, from concept to practice in EU regulations?
Let's 10 years might be optimistic, yeah.
Okay, I would, Greg.
Great, David, thank you for a wonderful talk and for taking us forward, and your comments at the end about gene environment and how that's going to lead to interventions and preventions. I'd like us to think about 10 years from now, 20 years from now, the day when sequencing is so cheap that we know everybody's genetic profile, and we've unraveled the exposome. We now know everything about our exposures. And at that point, how do we move towards this balance in terms of intervention and prevention? What do we do at the population level in terms of being able to things that are broad exposures that impact us all, and what do we do with the individual level where it's more genetic susceptibility? So how are we actually going to be able to operationalize this exposome information so it comes back down to the individual? You said in your comments it was you can do this, you can do that. You're talking about individuals, but there's also governments as well. So how are we going to make use of all of this?
Yeah, sure. So it's a great question, and I'm not sure I have answers to it. There's not one answer, undoubtedly. I think ultimately where the impact is going to come is by informing individual behavior, and it's not going to be a government action. So you've already seen some examples. There was a poster in the competition on looking at different commuter behavior and route, and so you can inform an individual. If you want to reduce your exposure to particulate matter, take this route that takes you through a park as opposed to this route that takes you along a highly populated roadway. Once we have that next step of the integration of the genome and the exposome, and you're really able to tailor that message to the individual. So you are an asthmatic that has genetic markers that indicates that you are going to have enhanced susceptibility to ozone as opposed to particulate matter-induced asthma. And so let's really target your route and your personal intervention towards reducing your exposure to ozone as opposed to particulates. Whereas another individual may have greater susceptibility to some allergens. And so let's keep you away from the farms. So I think ultimately the impact of this is going to be informing the individual about what are their risks, and what how can they use their understanding of the exposome to improve that. I think Dennis has a follow-up on that, maybe.
I was just going to say, we actually are in the break time, so not to get you too far behind in the line to get coffee, I would actually suggest that we table the discussion for now and move to the break. And then see you back at 11 o'clock when we actually continue. There will be a panel discussion at the end of the day, so a lot of these questions actually can be raised at that time as well. So thank you very much for all the speakers this morning, and hope to see you in half an hour. Thank you.